So the previous video in Lecture 9 looked at what controls a disease within a person, or what controls the severity or the likelihood. And now we're going to look at factors that control transmission and occurrence of diseases. And this is epidemiology. Epidemiology is a big field that includes really any kind of public health stuff that can be studied. So I looked at going into epidemiology when I was an undergraduate, and I read some epidemiology journals, and I was surprised. I kept finding articles about things like roller skating injuries. Because what epidemiologists do is they figure out what risk factors are. And they're trying to figure out, is roller skating a risk factor for X, Y, and Z injuries? The reason they have to do that is because there might be like insurance companies that want the data. You never know. And so the more attention-grabbing epidemiology is when we have an outbreak of a disease like COVID, or maybe more famously like Ebola, where the disease is scary and we want to know how to predict what it's going to do. And political leaders need to be able to make decisions based on predictions of what will happen if we respond in one way or another way. For our purposes, we are mostly interested in infectious diseases and how they spread. And what we would study, the questions we would try to answer would be the etiology. So what is actually causing it? What organism is causing it? How do you describe the people who get it? Are they old? Are they young? Etc. How is it spread? That is huge. Is it spread via droplets or aerosols or the fecal-oral route and things like that? So etiology is pretty simple. Koch's postulates can help us nail down what organism causes the disease. If Koch's postulates don't work, we have experience in dealing with it in other ways. Demographics is a little bit more complicated. We can describe a lot of different traits a person could have, a lot of different groups they could belong to, such as, again, young, old, rich, poor, etc. And a lot of those things can correlate to the risk of getting a disease. So some of the things we want to think about with demographics are really important words you need to know. Let's get into it. So incidence is the number of people being infected in a given time. So like the number of people testing positive for COVID in a given day. And you can compare a day during a surge to a day at another time, and you'd see the difference. And that's how you use incidence numbers. With prevalence, what you're thinking about is how many people have the infection or how many people are getting over the infection. So prevalence would be like the cumulative number of cases of COVID. Before COVID, one of the things that we've been trying to publicize is HIV, because it is a global, we're still in the middle of the HIV pandemic. That has not gone away and won't go away. We don't think about it as much because we have a lot of infrastructure in the US for treating HIV and preventing it from getting to AIDS, the acquired immunity deficiency syndrome. But outside of the US, it's still a major problem. So if so these numbers we can get do help us understand a lot about a disease. So from the incidence, we can figure out whether the disease is spreading faster or slower at a given time. And from prevalence, we can see how many people are affected and how much of a burden is it on society, things like that. So some words you should know are things like sporadic and endemic. Sporadic diseases show up occasionally, and we don't. A good example for us would be like coccidiomycosis or one of those fungal diseases. they don't go away, but most people don't experience them. An endemic disease is one that's always present in low levels and never goes away. So the common cold is the best example of this, where if you are currently alive, you have a non-zero chance of getting the common cold any day, any season, whenever. There's a chance of getting the common cold wherever you are in the world. But it doesn't happen in big outbreaks. An outbreak is where we see words like epidemic. Epidemic is a tricky word because it is not defined in the same way for every disease, and it's not defined in the same way in every place. So every public health department for a county or a city or a state or a nation has certain data they collect from hospitals, and it depends on what kind of laws have been passed to let them collect data. And so they have to declare an epidemic of a certain disease if the data show some threshold. And the data they would get for something like HIV would be different from the data they'd get for something like influenza or COVID. For quickly spreading respiratory diseases, they might be looking at indirect data, like the number of people with respiratory problems showing up at hospitals in a given day. So the incidence of respiratory problems. But for other diseases, they would have specific tests, and they would be defining it based on number of positive tests. But in general, an epidemic is used to describe a local or countrywide outbreak of something in a region. So epidemics historically would be like the HIV epidemic in rural Indiana about 10 years ago, and the human papillomavirus in the US. Those are examples of epidemics. And then pandemics we think about a lot now because COVID is the biggest, most noticeable pandemic of our lives, certainly. And that's a global outbreak of a disease. So if it's affecting multiple countries at the same time, it's a pandemic. That's all it means. We are in the midst, again, of an HIV pandemic. We're in the midst of the COVID pandemic. There have been influenza pandemics roughly every 10 years, and some of them are enough to really change how society behaves, and some aren't. So, for example, the H1N1 in 2009, that influenza in some places, people didn't really notice it. In hospitals, they did, because higher numbers of young people were dying of respiratory diseases. And my first group of students were in Wisconsin, and their high schools had closed during that because so many students had had influenza. But there were no lockdowns, there were no mask orders, because it wasn't spreading fast enough to threaten all of society, and people could avoid it for the most part. And you could contrast that with the 1918 influenza epidemic, or pandemic, sorry, which is very similar to COVID. The same things happened in society. So there were mask mandates and anti-mask activists all over the country. There were people who didn't believe that the disease was happening, and others who spent day after day after day treating sick people. So pandemics can range, again, from something that a typical person wouldn't notice to something that a typical person could never ignore. Emerging infectious diseases is something we have to think about a lot. These would be new diseases. COVID is a great example. It didn't exist 2 years ago. Other examples could be when a pathogen changes. If it gains antibiotic resistance or something like that, it becomes it causes a new disease. Also, again, with COVID and with influenza, when these viruses make a jump from one animal to another, like for COVID, from probably bats to humans, or from influenza from birds or pigs to humans, that's when a new strain appears. And so that's an emerging disease. We can also think about, and we have to think about how diseases are transmitted, so how pathogens move from person to person, or from one place to another. And so if we think about transmission from one person to another, it can be direct, so that's person to person, like typically we think of with something like COVID. It could be indirect, where something is contaminated, like drinking water in the case of cholera, or a contaminated food in the case of a lot of foodborne outbreaks, and then something like droplets, and that's a special case. There are different ways that disease transmission can happen. A person can directly contact an uninfected person. You can imagine a sexually transmitted infection or something spread by touching a person. And we could think about this happening at any time during an illness. So during incubation or the prodrome. And so just to review, I assume you've seen this before. When you first get infected, before any symptoms or signs appear, that's the incubation period. And the prodrome or prodromal period is when the first non-specific signs and symptoms appear. And these would usually be the response of the innate immune response. You'd notice the innate immune response. And then the main illness itself is when you get the recognizable signs and symptoms of a disease. So for something like COVID, there's several days between when we're exposed and when we start noticing the general unpleasantness. We start noticing loss of smell and coughing and all that stuff. That would be the illness. And then depending on the disease, there can be a separate period of decline when the immune system is starting to successfully fight off the pathogen so that its numbers decrease. And then the convalescence, when the person slowly heals from whatever damage they had, either due to their own immune response or directly from the pathogen. Some other things to think about would include reservoirs. So these would be where you can expect a disease to keep coming from. And there are a lot of different types of reservoirs. So, for example, human reservoirs would be carriers. You can have carriers who are infected, but they're not having signs and symptoms, but eventually they will. So HIV is a great example of this. The reason HIV could spread so quickly through a population was that there wouldn't really be any distinct signs and symptoms of it. Nothing really diagnostic that a person would notice for 5 years or something like that before AIDS would start. And so a person could keep spreading HIV without knowing. There can be chronic carriers who have like a many years long or lifelong infection, and for whatever reason their body is able to prevent it from getting worse. So they don't have signs or symptoms, but they keep shedding the pathogen. A clear example of this would be like typhoid Mary, if you've heard of that. A person whose gallbladder was infected with Salmonella typhi, which causes typhoid fever, which is a fatal or a very dangerous disease. And since she never had symptoms of it, she didn't believe she could cause it. And so she worked as a cook in several different households, and everywhere she went, some of the people who ate her food would die. So that's a really glaring example of a chronic carrier. In cases where animals are the reservoirs, those are called zoonoses, and a good example of this would be rabies, where it can really infect any mammal, but there are particular animals that tend to give it to us. So outside of the U.S., dogs are by far the biggest spreaders of rabies, but since we can vaccinate dogs against it, we don't typically get it. There are a lot of different diseases that people who work with livestock have to think about, and veterinarians have to be vaccinated against a lot of different pathogens, because different animals can carry different pathogens that can jump to us. Vectors are a separate thing. So let's notice these two words, reservoir and vector. A reservoir is a kind of a long-term source of a disease. So a pathogen can continually be released from a reservoir. On the other hand, a vector is a short-term carrier, so to speak. So we could think about things like mosquitoes. Mosquitoes are arthropods, and a mosquito can transmit a pathogen from one person to another, but a mosquito doesn't live long enough to be a long-term source of that pathogen. So a vector is usually a short-lived animal, like a fly or a mosquito. There are other reservoirs for some diseases that are not alive, and so these are less common, but we could think of something like Clostridium that just lives in the soil. Most clostridial species live in anaerobic sediments, and eventually they can get into a person. We could think of Legionella, which it can survive in fresh water, and it can infect amoebas that survive in fresh water in water tanks. And then if a person inhales that water, it infects their lungs. And so in that case, fresh water really is the reservoir for that pathogen. In these cases, people are not spreading Clostridium or Legionella from person to person. They really are coming from the environment. One of the things we think about with like indirect transmission is fomites. Fomites are physical objects that get contaminated and transmitted pathogen from one person to another. So an example would be a doorknob. And some infectious diseases can be transmitted by doorknobs and others cannot. So Salmonella typhi, again, causes typhoid fever, is a good example of a disease where a person who has it will get it on their fingers and spread it to whatever they touch. And it doesn't take much to infect the next person. Norovirus can be spread through fomites. Definitely something to think about if you work in a clinic. So respiratory droplets are emitted from usually our mouth or nose, and they are small drops of water or saliva that contain within them whatever microorganisms were present in that water or saliva. And so this picture shows a person sneezing. There's a light source back here and a dark background. So you can see, hopefully, large objects. These would be large respiratory droplets. And so what these would do is fall to the ground because of gravity. What we can also see is a kind of a purple or bluish haze, and that is caused by light scattering from microscopic droplets. These will not fall to the ground nearly as fast. So while these big droplets might fall from the air in half a second, these might take more like 4 hours. And so depending on which of these is more likely to be a good home for the virion, you could have very different disease spread patterns. So SARS-CoV-2, which causes COVID-19, mostly seems to spread through these large droplets. So people are most at risk of getting COVID-19 if they are face-to-face talking with someone who is infected, because that person, their large droplets can cross that distance. What we don't know is how often do people get it from fine aerosols that stay in the air. For diseases like measles, where the aerosols are how people get it, or norovirus, a room can be contaminated. The air in a room can be contaminated for hours after the infected person leaves. So someone else who walks into the room and breathes is exposed. Most of the time, it seems like COVID is not spread this way, but there is evidence that the super spreader events, such as the famous wedding in New England, and a few other major events, have one infected person causing new cases with people who were never within 6 feet of them. So one person can spread it to people on the far side of the room if tiny aerosol droplets are the way they're spreading it. So again, these big droplets do not travel far. The small aerosols do travel far. And airborne transmission is a specific thing. So big droplets aren't considered airborne because they fall out of the air. Airborne droplets are the ones that stay in the air for a long time. And again, with COVID, these seem to almost to not usually be the way COVID is spread. So let's think about how small one of these particles can be. The largest it can be is 5 micrograms, and that's the mass of 5 microliters of water, and that is a droplet that you could just barely see. Most of them would be much smaller than that. The smaller the droplet is, the longer it stays airborne. So the microscopic ones are the ones that stay airborne for hours, and if there are breezes in the room, those air currents will keep lifting the droplets back into the air. One thing you should know about this is that humidity controls droplet size. So humid environments are going to make larger droplets because the water from the humidity will condense onto the particles and make them bigger, and so they will fall out of the air faster. In dry environments, the droplets will shrink because of evaporation, and they will then stay in the air longer. So in dry environments, we would expect respiratory droplets to stay in the air for a longer amount of time. That's one reason why influenza and the common cold spread more quickly in the winter in enclosed spaces. The other thing I want to mention is, like, why don't we know how COVID spreads? Why can I not give you a confident answer? And the reason for that is because we can't do experiments with COVID. All we can do is look at public health data and try to figure out what happened. For diseases like the common cold or influenza, we can get volunteers and spray them in the face with these viruses. We can do that safely because a healthy 20-year-old volunteer is unlikely to have severe complications of these diseases. They're not going to have complications of the common cold, or any complications they have we can treat. And influenza we can directly treat with antiviral medications. We can't do that for COVID. We cannot stop a COVID infection with any drugs that currently exist. What that means is that if you spray a volunteer in the face with COVID, if they're unlucky, their immune system won't control it and they'll be dead in a month. So we can't do volunteer exposure experiments with COVID, and we won't know how it spreads with that much confidence until we can do those experiments, or until we have years and years and years of well-analyzed observational data. So public health people, epidemiologists, do great work doing these observational studies where they figure out what happened in a room and they take what conclusions we can. But it's not the same thing as being able to do a controlled experiment. Controlled experiments give us a lot of confidence in our data. So that's all I'm going to say about that. The last two slides here are about the portals of entry and exit. So how do pathogens get into us? Well, the respiratory tract is the main way. By far the most common infectious diseases are the common cold, different strains of the common cold, and they're spread through the respiratory tract. It's when the virions can touch the epithelia in our upper respiratory tract, that's when the infection starts. Very notable other pathogens affect our guts. So fecal-oral route of transmission, where we defecate and release large quantities of pathogens and then somehow contaminate the environment, and someone else somehow gets those pathogens into their mouth. That's how gastrointestinal infections are passed along. Clear examples include norovirus. A person with norovirus will vomit and have diarrhea. And both of those processes generate aerosols. And that means that a person with norovirus can enter a room and contaminate it for many hours. And all it takes is a small number of those virions, like 10, entering a person's digestive tract to infect them. Less common would be sexually transmitted infections or other infections of the urogenital tract, but they obviously exist. And then a thing to remember with something like respiratory tract is that the eyes, lacrimal fluid drains to the respiratory tract. So the eyes are a route of possible infection. So if you want to avoid COVID, wear a mask and wear safety glasses or goggles or a face shield. That protects your eyes as a route of infection. Less commonly, organisms can get into our skin. There are some really nasty pathogens that if they land on our skin, they can get in, but most cannot do that. And then lots of microorganisms can make us sick if they get in through breaks in the skin, as we will see. And then we can imagine lots of different ways microorganisms can come out of the body. So, some of these slides have been laundry lists of words and things that are not going to surprise you. And I hope what you picked up on is I spent more time talking about things I thought might surprise you. And what you need to do with these videos is just make sure it makes sense to you. Make sure you understand what all the words are, so that when I use the words on an exam, you can tell which, you can match a word to its definition, etc. I will see you later.